Welcome everybody again to another episode of the IdeaXme show that profiles humans behind the big ideas that are shaping our world, inspiring the future and future creators, and for all those really great stories. I'm Ira Pastor, your life sciences ambassador along for the journey today. So as viewers of the show know, a few months ago we had a thought leader on from the Google Alphabet portfolio of companies, the Verily, discussing with us everything that they were doing on the sort of the moonshot health front in regards to medical devices and wearables and surgical robotics and things of this nature. Today we have another fascinating guest from within this family of companies, specifically from the Calico Life Sciences organization, which is a research and development biotech company that's backed by Google, ultimately with the goal of combating aging and age-related associated diseases. Dr. Adam Freund is joining us. He is a principal investigator from Calico. His lab is focused on studying aging at the systems level in mammalian models, and ultimately works to identify and test post-developmental interventions to either slow or reverse aspects of aging, as well as understanding whether aging is regulated by similar mechanisms across various tissues. Dr. Freund has a bachelor's in material science engineering from Stanford, a PhD in molecular and cell biology from UC Berkeley, and has studied aspects of biology of aging throughout his career. He did graduate work in Judy Campisi's lab at University of California. He was at the Buck Institute, focusing on the regulation of inflammatory networks during cellular senescence. Did postdoc work with Steve Artandi at Stanford, where he developed high-content screening methods to identify regulators of telomerase. A lot of really exciting topics here. Adam, thanks for taking the time out of your schedule to come on the show for a little while.
All right, thanks for having me.
Yeah, it's a real pleasure. Typically, we start things off by handing our guests the floor for a little bit. If you could just take a few minutes to introduce yourself, sort of, going back to the basics of, where did you grow up, how you got interested in science and molecular biology, aging, and ultimately a little bit of your career journey to date, both in academia and industry. That'd be a really great introduction.
Absolutely. So starting from the beginning, I grew up in Texas, and I came out to California for my undergrad at Stanford, and I've been here ever since. And so how did I get into science and aging more directly? Well, I would say from a young age, I was drawn to pretty impossible problems. Not necessarily because I like to fail over and over again, but just because I always came from a position of what if, right? I was always asking, well, what if that were possible, what if that were true? And I grew up on a pretty steady diet of Star Trek and science fiction novels. And so
Excellent.
I was always exposed to these possibilities. And I actually didn't start being interested in aging. I started being interested in space travel, probably from some of these stories. And it seemed to me that the rate limiting step in making any of these stories a reality was always, faster than light travel. That was the thing. If you had that, you could make it work. And if you didn't, there really wasn't much of a path forward. And so I actually held on to the idea that I wanted to be an aerospace engineer all the way through my freshman year of college. And at that point, I had some frank conversations with a few professors in a variety of departments, and they made it very clear that not only was there not a lot of funding available for faster than light travel projects, there also wasn't a lot of much future in trying to break physical laws. And so I decided to switch my field of focus, and I moved into the material science department, where I really got interested in nanotechnology and the possibilities there. But once again, I think I came across the reality that a lot of the promise of nanotechnology, the sort of science fiction-like promise of nanotechnology, is perhaps in violation of certain physical principles. And again, not necessarily a huge path forward there. And so I decided to move yet again, and near the end of my undergraduate career, still in the material science department, I decided to focus on biology and the applications of material science and nanotechnology to biology. And that got me really interested in aging, because as you sort of explore the field of biology, there's so many problems to choose from, right? So much is not well understood.
Sure.
And so many different diseases that one could choose to dedicate one's life to. And if you take the utilitarian view, aging is sort of the biggest problem of all. And this was a big step for me. This was a big step in the right direction versus something like developing hyperdrive, because at least at face value, it's at least physically possible, right? There's nothing about delaying or reversing our biological decline that breaks any physical laws. It's on the extreme end, I think, sure, entropy will always win, but every living thing on the planet is an example of locally defeating entropy, at least for a time. So for practical purposes, it's possible. It's just a matter of understanding how to intervene, what to target, when to target it, where to target it. So really a matter of untangling the system. And so once I focused in on aging, I decided that I would pursue that, and haven't changed my mind up to this point. And so I went from my undergrad at Stanford to my PhD at Berkeley, studying molecular and cell biology with Judy Campisi. And in her lab, really got into the field deeply. First in cell senescence, and then I went into my postdoc studying telomerase, which is a enzyme that actually regulates sort of how the ends of chromosomes shorten with every division, and so has some potential relation to aging. And I did that with Stephen Arcandi at Stanford. And then at the end of my postdoc, was offered an opportunity by Cynthia Kenyon, who is well known in the aging field, to join a startup, Calico, that Google had just created. And this was a dream for me. I mean, the idea that I could continue to study aging, but do so in a setting where there was an eye towards drug discovery and development of therapeutics was really exciting, and remains really exciting. And so I jumped at that chance, and I've been a principal investigator there ever since.
Wonderful, wonderful. I appreciate that background. You last year you wrote a paper. It was in the journal Cell Systems. It was entitled "Untangling Aging Using Dynamic Organism-Level Phenotypic Networks." And you talk about some very interesting challenges, in the sense that we have all these very interesting molecular and cellular targets available to us when we think sort of the bottom-up way that sort of we develop drugs and so forth today. But also from a top-down perspective, a lot of these phenotypic changes that are occurring, we don't know if they represent aging or not. And you propose very interesting strategy sort of to the merger of these two, using some of the themes that we've discussed on the show in the past, in terms of sort of the systems biology folks looking at sort of the multidimensionality of how aging unfolds and so forth, using a lot of these different inputs. Can you take us on a little walk through some of your vision in this context of sort of the systems, a perspective of aging, and how not all aging looks the same? Just like we learned that cancer is hundreds of different things potentially, that aging may have some very unique properties to it that it's not always one simple thing, but we can approach things with this systems biology, sort of multidimensionality, the state space concepts to further understand it. Walk us through this a little bit, if you would.
I'd be happy to. And you're very kind to call that paper sort of a nice systems biology perspective, because really, I mean, I'm not a card-carrying systems biologist by any stretch of the imagination. This is something that I sort of fell into mostly by simply sitting around in my office and talking to other people at Calico and thinking about a key problem. And that problem was, if we are an aging company, a company trying to develop drugs for aging, how would we know if we had an anti-aging drug? And for that purpose, I mean something that delays aging or reverses aging. What would be the metric by which we would decide to pursue a target and a pharmacological intervention? And you say, well, okay, that's pretty easy. It'll delay or it will reverse aging, and it'll do it in some models, and then it'll do it in some higher models, and eventually we'll run a clinical trial. And I think the problem is, it's not always clear what delaying or reversing aging looks like, even in model organisms.
Sure.
And so my colleagues and I really puzzled over that for a long time, of what does success look like here? And I think the gold standard in the field, of course, is often lifespan. And at a certain level, I think lifespan works really well. Take C. elegans, right? A worm, a small, short-lived worm, lives for a couple of weeks. And there are interventions in worms that can extend lifespan by threefold or fourfold. I think there are combinations that can even extend it sixfold. And in that case, there's not really any question that you've fundamentally modified the nature of decline in virtually every individual in that population.
Sure.
But when we move to mammals, and take mice as an example, I mean, there's plenty of studies out there that show a 5, a 10, a 15% increase in lifespan across a population of mice. And it's not always clear what that means. It's not always clear that some concept of general decline has been prevented. It could be that a certain cause of death was ameliorated, or perhaps only in a subset of the animals did something happen. But since you're looking at a population readout, it becomes a little bit hard to understand what's going on. And so we got into this idea that aging really is a set of problems that develops in each of us that we'd like to avoid. And if that's the case, then we want to measure those problems at an individual level. We want to measure health, as it were, because if we can understand how health declines and we can measure it robustly on an individual level, then we really have a platform through which to test interventions to see if they can delay that decline or even give health back in the concept of reversing aging. And so that got us a little bit clearer towards a solution. But again, we were like, well, what aspects of health do we want to measure? I mean, there's almost an infinite variety of things that one could measure. And there's always a conversation about what constitutes aging versus the effects of aging or diseases like age-related diseases. And much ink has been spilled on this problem.
Oh, yeah.
I think it gets largely semantic and it gets largely philosophical. But it occurred to me and to some others at Calico that if take a human clinical trial, if a drug was able to delay the onset of the top 10 age-related diseases that exist, right? So cancer, Alzheimer's, neurodegenerative diseases, stroke, cardiovascular, kidney problems, that kind of thing. And that's all the information that we had about that drug, was simply that it delayed the onset of a variety of age-related diseases. Would that be sufficient for us to call that thing an anti-aging drug? Well, you can decide for yourself, but I'll put it this way. I would certainly be interested in pushing that drug forward, right? I think that it would clearly extend the health span of a lot of people in our population. And that makes it inherently more valuable than a drug that extends the health of a smaller subset of people or extends it for less long.
Sure.
And so what it really came down to for us was almost a counting of deficit type problem, in that we want to be looking at a variety of health endpoints. And it's not that we're going to draw some arbitrary cutoff and say, only when we've affected this endpoint or this percentage of endpoints have we affected quote unquote aging. It's more that the more things that we affect, the more health deficits that we can prevent from occurring or delay from occurring in a larger number of individuals in a population, whether that population is mice, humans, or any other organism, the better. It's sort of a relative prioritization approach. And in that paper, what I try to do is lay out the idea that in different populations, those health deficits might be different. And that is a reality that we have to face. It's not one that it's pleasant to, I think, really think about, but the reality is there's no guarantee that the way that a worm breaks down is the same as the way a fly breaks down, is the same as the way a mouse breaks down or a human breaks down. Sure. Just because all things break down doesn't mean they all break down from the same set of causes. And that's a hypothesis, and it's one that we can test. And I think there's a lot of empirical evidence out there that certain mechanisms are conserved, but it doesn't mean that all mechanisms are conserved. Sure. And so one of the things that we think a lot about is how to increase the odds of translatability. And this isn't something that's unique to aging. This is something that every drug development company goes through in trying to bring a drug to the clinic. You have your preclinical models. You're trying to understand what's translatable, what's not. And so we're working on developing a variety of tools to do that. But it all starts from being able to robustly measure the decline in health that occurs in our preclinical models.
Then working along those lines a little bit, you a couple weeks ago, I watched you. You gave this presentation at the Aging Research and Drug Discovery meeting. Your focus was inhibition of the longevity regulator, PAPPA, and modulating tissue homeostasis. this stands for pregnancy-associated plasma protein A, which is an interesting protease that binds with insulin-like growth factor, which also has a history in sort of the longevity space. I gave the big presentation there. I was wondering if you could walk us through just some of the highlights of this, because really interesting presentation, and sort of if you could also give us just a little background, because in looking into this protein, it's involved in a lot of other stuff you wouldn't really think of in terms of aging, in terms of the structure of the placenta and intermediate growth. Take us on sort of how this concept started and how it got to the point of, wow, this may be involved via this IGF mechanism in homeostasis in other tissues.
Absolutely, it's a good question. So the way that we, at a high level, the way we got onto this target was in thinking through from first principles the problems of developing drugs for aging. I think in your previous question, I touched on, well, how do we measure aging so that we can assess putative interventions? But the other side of that, of the problem is, well, how do we identify putative interventions in the first place? Where do we come up with the ideas?
Sure.
And I think that when we started Calico, a lot of us didn't come from a background of studying aging or having targets in mind when we joined.
Sure.
And so we perused the literature. And Cynthia Kenyon actually put together who of course was at Calico at this time, put together a really nice sort of symposium of investigators who had mouse models of extended longevity. Mm-hmm. And all of these various investigators came together in San Francisco. This was back in 2014, and talked through their models. And it was a really nice, intimate setting. We were able to dive into the similarities and differences. And one of the models that was presented there by Cheryl Conover, who's an investigator at the Mayo Clinic and who has done the work around PAP-A, was the PAP-A knockout mouse. And the PAP-A knockout mouse lived something like 30% longer, and she and others had done a great amount of work characterizing that it had all sorts of delayed age-related pathology, so not just lifespan, but also health span. And it was implicated in regulating IGF signaling. And this was interesting to us because IGF signaling, of course, is, in my opinion, and some disagree, but I think the most validated anti-aging intervention that we know of.
Right.
And I say that because it is conserved. It was really the first mechanism identified by Cynthia Kenyon in worms, the NAF2 mutation many decades ago.
Right.
And it extends lifespan of worms, of flies, of mice, and there is evidence as well in people that inhibiting this pathway can delay aspects of aging. And the effect sizes, at least in these lower model organisms, are huge. In the worm, it's a single mutation, and it more than doubles the lifespan of the animals.
Sure.
And in mice, you have things like 30% extensions of lifespan, which is not as good as a worm, but it's sure a lot better than much else that we have. And so it was always interesting to us that IGF signaling was sort of just sitting there as a low-hanging fruit, but it's not an easy pathway to target. And the reason for that is that, first of all, there's a lot of regulators. You have IGF, which is the ligand itself. Then you have multiple receptors. There's also a second ligand, IGF-2. There's IGF binding proteins that regulate the bioavailability of IGF in different tissues and contexts. Very difficult to untangle. And I think one of the things the field has struggled with is understanding when and where IGF signaling is happening. And this actually, there were a number of pharmaceutical companies that tried to develop IGF-1 receptor inhibitors for cancer about a decade or so ago. And one of the things, and they eventually didn't make it to the clinic, and one of the reasons that people postulate that happened was a lack of good understanding of a lack of biomarkers for the pathway, a lack of understanding when the pathway was actually being modulated. The other problem is that there are really, there are feedback loops that are highly tuned, so that when you try to inhibit IGF signaling in one place, it's the hypothalamic pituitary axis. Your body upregulates IGF signaling through the brain, and that causes a big spike in circulating levels of these proteins. And so sort of paradoxically, by trying to inhibit IGF in one tissue, you could end up with a lot more IGF signaling in other tissues or something like that. And so this was all very complicated and confusing to us. But PAP-A gave us perhaps a unique lever around which to intervene on this pathway. Because what's interesting about PAP-A knockout mice is that they have reduced IGF signaling, but without altering circulating levels of any of these factors. And that's really important in trying to untangle the mechanism and in trying to have a precise intervention. And so how does this work? So PAP-A is, as you mentioned, it's a protease, so it cleaves other proteins. And the proteins that it cleaves are a select few of these IGF binding proteins, namely IGF binding protein 4. And this releases IGF to signal through its receptor. And this only happens in places where you have all of those factors present. So you have to have PAP-A, you have to have IGF-4, you have to have IGF-1, you have to have the receptor. And this only occurs in a subset of tissues. And so this allows IGF signaling to be modulated with spatial precision, and if you have a pharmaceutical, with temporal precision. Now, there's no guarantee that this, wherever PAP-A happens to be expressed and active, that this would be the location that would modulate aging. But the PAP-A knockout mouse suggests that it is, right? Because you have about 30, as I said, about 30% extension of lifespan and health span, which is about as big as anything we've seen in mice by inhibiting this pathway. And so, although we don't fully understand biology, we think that we have relevant biology with this target. Now, you made a good point that it stands for pregnancy-associated plasma protein A, and it wasn't discovered initially as an IGF regulator or an aging factor. It was discovered in the profiling of human pregnancy serum. So the blood from pregnant women, they didn't even know what it was initially. They just knew that this factor increased something like 10,000-fold. And it was later identified as this protease, the cleaved IGF binding proteins. And the thinking is that in humans, but interestingly not in mice, it is regulating fetal growth through the placenta. And it does that by regulating IGF-2. And this, as I said, this seems to be really a unique feature of primate regulation of PAP-A. Okay. And to the best of our knowledge, is somewhat independent from its role in aging. Because as we said, in mice where this pregnancy increase doesn't occur, we see a large extension of health span and lifespan when we inhibit or delete the protein. And so I think that there's a lot of interesting biology around PAP-A and pregnancy. But for, I think, the purposes of aging, we're thinking of it as something that might be somewhat independent. And the other reason that we think that is that in a lot of really great work from a lot of other people, they found that although PAP-A is increased in pregnancy something like 10,000-fold, it's 99% or higher inactive, bound to this endogenous inhibitor. And so why that occurs, again, we don't really know, but we don't think that it's potentiating IGF signaling as much as you might think simply by the expression levels. And so we got interested in this, and we decided that we would develop an antibody against PAP-A that would neutralize its activity. And we did that, and we did some really just early stage experiments to validate the target. And this is something we always do, and especially when they're targets that we don't know anything about. I had never personally worked on PAP-A before. I was really just pulling it out of the literature, and the literature looked solid, but as I'm sure you know, these things always have to be verified. And we were really happy to see that we were able to reproduce a large number of the seminal findings. But the biochemistry played out. It was indeed a protease that cleaved these IGF binding proteins. It did indeed potentiate IGF signaling. And then we did some initial in vivo experiments to validate some of the physiological effects of inhibiting PAP-A in a variety of contexts. And we were able to reproduce those as well. And so this gave us a lot of confidence moving forward. And so now we are developing inhibitors of PAP-A for a number of indications.
You know, prior to Calico, in your time in academia, you were involved in what I call sort of a lot of the hot areas of aging in terms of the telomeres, in terms of the inflammatory secretome of senescent cells, things of this nature. obviously, these things start off hot, and they are hot, but we learn that, there's a lot of balance per systems biology that's required. Too long telomeres, too short telomeres, not always good. Senescence is bad in the long term, but it has benefits in wound healing and other purposes. I'm just interested in, from the time you spent on some of these really popular aging targets in academia, now moving into industry, have there been any interesting insights, either with regard to the telomeres or senescence, that you think, using some of your new thinking in terms of the systems approach, that you could talk about, beyond what we see in the press with regard to these two interesting areas?
Well, I can only give you my sort of personal thoughts about these areas. And certainly nowadays, there are people who know a lot more about senescence than I do. I would say that at Calico, we have largely not delved too deeply into that particular area, not because we think there's anything wrong with it, but because it's so well covered by excellent scientists in both academia and other biotechnology companies. And our goal is always to fill gaps that we see, right, that aren't being addressed by others, not simply to race to the finish. I think we want to be a piece of this puzzle that contributes to the greater good, not one that's driving unnecessary competition. And so, so I will say, but I'll say what I can about my thoughts on senescence and telomeres based on my limited knowledge.
Please.
For telomeres, I think it's a bit, in my mind, more straightforward. I think there's no question that telomeres and telomerase are an incredibly important biological process, biological enzyme. The regulation of the ends of chromosomes is fundamental. And we can see that when it goes wrong, certain things happen. And we're lucky in a way that there are actually examples of humans with mutations in these pathways and these proteins, because it shows us what happens when telomerase regulation goes awry. And on the activating side, what happens is cancer. And this was shown very clearly in a variety of families who have mutations in the catalytic piece of telomerase, and they get melanoma, familial melanoma, as well as a variety of others, but melanoma appears to be the most prevalent type of cancer that arises for reasons that I think are totally clear. When it's broken, there are families who have mutations in telomerase and other regulators around telomerase, and they develop a disease called dyskeratosis congenita, which is a failure of proliferative tissues, largely. And this makes some sense. The proliferative tissues are the ones where the telomeres shorten fastest, because telomeres shorten with every cell division. And so the more you have a tissue that divides, the faster the telomeres shorten. And when they shorten too much, those cells either die or they senesce, and that leads to tissue level problems. What's notable, though, I think, is that these diseases, especially on the dyskeratosis congenita side, don't really look like what we would call normal aging. There's bone marrow failure, there's leukodystrophies, there's other sort of strange phenotypes that we don't generally associate with aging. And there are many reasons why that could be true, and telomeres could still be involved in normal aging. There's differences, of course, between germline mutations and the shortening of telomeres over an entire lifespan. It doesn't have to be that there's a disconnect here. But looking at just the most parsimonious explanation, to me, it seems that it's likely that telomere shortening does regulate and does play a role in certain pathologies, but I am, on a sort of relative prioritization scale, less convinced that it plays a role in normal aging. I think one age-related disease where it does seem to play a role is idiopathic pulmonary fibrosis. Because people who have mutations in telomerase develop idiopathic pulmonary fibrosis, right? We have the genetic evidence in front of us. And so it's not that it's not important, it's just that it's, if we're looking for these things that affect the functional decline across multiple tissues and mechanisms that are conserved across species so that we can study them easily in our preclinical models, telomerase has, there are some issues with it, right? Mice, of course, inbred lab mice have super long telomeres, and yet still age in a way that we think is relevant to human aging, and that that raises some concerns about how we can study this mechanism effectively. So I think that's where telomerase is. With regard to cellular senescence, of course, cell senescence can be an outcome of telomere shortening, but senescence can arise in a variety of ways, both in vitro and in vivo. And it's not clear how much of senescence in vivo is a result of telomere shortening, especially when you see it in mice where they start with these very long telomeres. And cell senescence it's a fascinating area of biology. And I think that what that field has really shown us, and this is something that 10 years ago, I would not have guessed, is that you can eliminate non-cancerous cells, that is senescent cells, in a living organism. And cells like something like 1% of a tissue or something like that. These are the estimates how many senescent cells are present in many tissues as we age. And that doesn't cause huge problems to the organism. In fact, it in many cases makes the organism better. And this is surprising at face value. I mean, if again 10 years ago you had told someone, listen, I'm going to clear out like 1% of your adipose tissue or your liver or your pancreas, and that's going to be good for you. And then these aren't cancer cells; these are just normal cells that have maybe gone a little bit awry. We're just going to get rid of them. I mean, you would get people saying things like, "You're going to turn organs into Swiss cheese." I mean, you're going to have lysis syndromes. You're going to have all sorts of problems. And I think what the senolytic field, that is senolytics being agents that kill senescent cells. Sure. What the senolytic field has taught us is that our gut reaction to this concept of pruning, if you will, non-cancerous cells in vivo, it is probably wrong. I think that we're not right to immediately believe this is going to be deleterious. In fact, if we prune the right cells under the right context, it can be beneficial. And what excites me about the senolytic field, and obviously there's been some issues lately with clinical trials and the initial version of senolytics not quite panning out. But to me, that's okay, right? Because what we have here is the first generation of an entirely new modality. And that modality is that we can go in periodically and eliminate the root cause of pathology. And that's so powerful for a variety of ways. One, it teaches us that we can target cells, not just pathways. Two, it gives us a therapeutic tool that doesn't have to be chronically administered. And when you think about aging therapeutics, that becomes very important, because imagine that you are trying to develop an aging drug and it has to be chronically administered because you're inhibiting a pathway, whatever that pathway may be. And so you say, okay, I'm going to go into 60-year-olds who are otherwise healthy, and I'm going to dose them with this thing for the rest of their lives. And that's going to cause a lot of benefit for them, and they're going to live a lot longer and have a longer health span, and that's great. But you're going into people without overt disease, as it's generally recognized. And thus the safety profile of one of these, of a drug like this, has to be incredibly clean. It basically has to have whistle clean tox if there's any downside to it. And I think that people who look into rapamycin have dealt with this, right? The immunosuppressive effects of rapamycin have been brought up a lot. Yep. And so people try to get around this by saying, well, we'll dose lower than the immunosuppressive effects, or we'll do cycles of rapamycin treatment. And it's the cycling that I think is particularly compelling to me, because and maybe with rapamycin, I think the cycles are a little bit closer together, but with senolytics, you can imagine that you go in once a year or so, and you dose and you clear out senescent cells, and maybe for a week after that, you do have some undesired side effects because you've accidentally killed off a bunch of platelets if you're taking AT263 or something like that. Right. But then those come back, and your senescent cells take longer to come back, and so you persist with this benefit without any active compound in your body for the next year, and then you do it again. I think that the tox profile that would be allowed by regulatory agencies for something like that could be, well, you could have a little bit more tox over a short period of time, and that could be okay if the benefits outweigh those costs. And so what excites me about the senolytic field is that I think we've just scratched the surface of what kinds of cells to target.
Mm-hmm.
But the promise of this modality is huge. And I really think that there's going to be a very long future. And maybe at some point we'll stop calling them senescent cells, and we'll start, being a little bit more perhaps precise with our language. Like, we're looking for chondrocytes that have this specific inflammatory profile.
Mm-hmm.
And at that point, much like aging itself, whether it's senescent or not becomes almost just a semantic problem. If you really understand the mechanism, understand the cells that you're targeting, that becomes the important factor, not exactly what you call them.
Absolutely. Absolutely. And I like your connection, sort of thinking forward towards, what the translation of these things is going to look like when you, as you mentioned, when you get to, I don't have any, I'm not really sick, but we see the benefit in clearing these things out, and what the, that's ultimately going to look like. I like when people start thinking about that ahead of time, because obviously that's coming, and it's going to look different in terms of compliance and things of this nature, and how these things are passed out and make it into the broader system. But it's, I appreciate that insight as well. Adam, at the beginning you mentioned, obviously someone with your background and showing up, Cynthia Kenyon and your job at Calico, dream come true, obviously a wonderful place for someone with your background. As an outsider, sort of looking at Calico, I come out of traditional big pharma. It looks very different, although you're working with AbbVie and other small biotech companies. Obviously, nothing confidential here, but can you talk about just sort of what it's like, sort of day to day, sort of like, I see there's. Basically, Calico shows a lot of folks like yourselves on their website, the different things you're involved with. I'm thinking sort of what it's like inside it. And although you're a biotech shop, you are an extremely well-funded biotech shop, more like a small pharma organization. What's the day-to-day like? You're not competing. It's not like you're the oncology team competing with the cardiovascular team for resources. You're all interested in aging, maybe some different approaches. But what's the day like for you? A typical day in the halls of Calico?
Yeah, that's a good question, Ira. And I recognize that there's a perception among some that Calico is a particularly secretive place, and that it's hard to know what's going on. And I would just say that it's far more straightforward than that. It really is just like many other places. We have hired people from academia, we've hired people from other biotech companies and pharma, and largely the culture is very similar to one that you find in a lot of other places. I think the perhaps a difference is that we are, by virtue of our setup with this funding from Google, and our view of aging being our ultimate goal, which is of course a very long-term thing,
mm-hmm.
we take a very long-term view.
Sure.
I think this is different from, it's perhaps more similar to larger pharmaceutical companies who really are thinking years, if not decades down the road, but it's much different from other biotech companies of our size. We're about 230 people right now.
Mm-hmm.
So usually a biotech company that size is burning through its phase 2 or its Series B funding, and really needs to push 1 to 2 products to the market as fast as possible. And we're in a different position.
Right.
And so what we're really focused on is spending the time to do the research, to build up targets that others probably wouldn't, because it's not always immediately obvious how to translate them. I think PAP-A is a great example of this. We chose it because we think that it plays a role in the fundamental aging process. But now what we need to do, and what we're doing actively, is trying to understand the mechanisms around how it does that, so that we can, one, convince ourselves it is a good target for aging, or convince ourselves that it's not, and two, think of a clinical development path. Because at the end of the day, we cannot actually run an aging trial right now. So we need to find some indication that we can use to bring PAP-A to the clinic, with the understanding and the belief that eventually we will be able to expand into other indications and other age-related diseases, if indeed it is an aging modulator.
Mm-hmm.
So we take the long view. So we do a lot of basic research. We do a lot of exploratory work. We also do a fair amount of drug development, and we have a lot of partnerships with other biotech companies, pharmaceutical companies like AbbVie, as you mentioned.
Mm-hmm.
We partner with very closely to develop most of our drugs, as well as academic investigators,
mm-hmm.
to identify targets that we view as low-hanging fruit, even if they are more specific to a single age-related disease like cancer or like cardiovascular disease. We don't have specific cardiovascular departments or things like that, but we are interested in wherever we see that we could provide benefit in the world of aging and age-related disease. And so some of our labs, and Calico is broken up into small labs on the research side of the organization, some of our labs are really focused on the fundamental mechanisms of aging. Some labs are more focused on specific problems. Some labs are focused on specific models, like using yeast to understand the basic principles of gene regulation and how that changes. And really, it's a very exciting place because there's such a wide distribution of research interests that there's always something new to learn. There's always someone interesting to talk to. And what we hope we do is combine the best parts of academia, this curiosity-driven, science-driven, exploratory state of being, with the best parts of industry, which is being goal-focused, having timelines, being very thorough about what we invest in, what we don't, validating our targets, and not being afraid to shut things down when they don't work. And so I think we're not perfect by any stretch of the imagination, but we hope that we can take the best parts of both of those fields and combine them into something special.
Oh, that's great. I appreciate that, and thank you for taking us for that walk through the secretive nature of Calico. That's great. I really appreciate that. One other quick question before I get to the wrap up. One of the things I noticed in your profile is that you are also, interestingly, a consultant for an equine biotech or DNA testing company. I'm always fascinated by the sort of the other areas that our guests are involved in or have been involved in. And I'm bringing this one up not from the perspective of sort of what you're doing in terms of DNA testing, but sort of this concept of larger animal models, as well as we had Ellen de Brebender, who used to run Marial's research group. She's now at Pepsi, but the animal health place is obviously extremely lucrative when you compare it to sort of big pharma. Big animal health is even more consolidated nowadays. I was just interested in maybe a couple minutes of what you do here, and do you think there's any interesting either large animal research that is relevant to what we do when we move beyond the mice and the rabbits and so forth, and then just in general, what you think of sort of I know that's not, a horse is not a companion animal, but some of these other areas within the animal health space may be relevant for aging research.
My journey to Edelon was an interesting one. I got involved in that company as a postdoc because they were interested in measuring telomere length in horses as part of a panel of assays that they were proposing to market to horse owners, as well as a series of genotyping tests, sort of like the 23andMe of horses. And by virtue of helping them do those initial assays, I sort of became involved in the company, became an advisor. But really, I think that my contribution there is really limited. I'm sort of tangentially aware of what's going on, but I think they're really focused on the genotyping phase of the company right now. And it seems they're doing quite well. It's really an exciting company. As you said, there's a lot of interest and value in understanding this, especially for horses, where the breeding times and the cycles are extremely expensive. And so the more that you can know about the kind of foals you can expect from a certain cross, the better. But really, it's completely separate from anything that I'm doing with Calico, and really completely separate from anything related to aging.
Okay, cool. I appreciate that, definitely. Just one other question. We typically we come back to you. Obviously, you've mentioned folks like Judy Campisi, like Dr. Kenyon. Obviously, you've run into, talked with a lot of fascinating thought leaders throughout your academic career, obviously in industry. You've Art Levinson leading Calico now, so a really nice mix of folks. Any other important influencers, mentors over the last decade or so that you may want to mention, shout out to at this point that, if it wasn't for them, Dr. Adam Freund would be, off working for Pfizer or something right now, as opposed to what you're doing?
Well, I have to start by giving another shout out to Judy Campisi.
Okay, sure.
Because she really took a chance on me when I was coming from a material science department. I had never done a biology lab prior to starting grad school. And I remember distinctly, I was convinced I wanted to join her lab because of the connection to aging. And I knew that I didn't have any of the necessary skills to do that. And so in our PhD program, we go through rotations where we rotate in several labs before we eventually choose one. And so I said, okay, I can't rotate in Judy's lab first, okay, because I will totally mess it up and she will never hire me. And so I actually, I spent the first half of my first year of grad school just learning the ropes in a couple of other labs. And I really, the PIs of those labs deserve shout outs as well, because they put up with me. And I really was experimentally kind of a mess at that point. But I learned, and I think by the time I got to Judy's lab, I at least knew which end of a pipette was, went into solution. That was good. But still, I really was very green. And she took a big chance on me, and there were postdocs in that lab who I'm still very close with today, who took a chance on me and taught me about senescence, about aging, and even more importantly, about how to do experimental biology correctly. And so without that, I really would not be where I am today. The other, I think, when I switched over to industry, as I said, Calico is in many ways like a research organization. And so a lot of the same processes and interactions that I had in academia happen at Calico as well. But there's also this drug development side of the organization, and there's thinking about high-level strategy and how to prioritize activities, where even a place like Calico, which is well-funded, time and money are not limitless. Sure. And we have to find ways of having really clear-eyed, defensible metrics to decide what to do with our time, both within labs and across the company as a whole. And watching how the leadership at Calico navigates that and manages to have a thoughtful and measured approach that both is exceptionally science-driven, but also really people-driven, and not the kind of thing that is entirely, that remains personal, and that remains just a really nice blend of different priorities and different sort of draws on decision-making. It's, I think, Art being a key example of this, but also the people that he's surrounded himself with. Hal Barron initially, who's now at GSK, but was instrumental in the first part of Calico. David Botstein, Cynthia Kenyon, Jeff Settleman, who's now at Pfizer, but again was there at the beginning. Nick Van Bruggen, others, Bob Cohen. A lot of these people were from Genentech, and not surprisingly, I think a lot of the good parts of Genentech have come to Calico. But what they represent are really modern business leaders, that almost servant leadership type mentality that I really strive to emulate, and it's really taught me a lot about how to navigate in a place where we work on such hard problems, and we're always taking these giant risks when we think about which targets to focus on, and how to do that and how to do that well is a true skill, and one that I have learned a lot about from them and hope to continually improve on myself. So.
Wonderful. I appreciate that last message. And it's been extremely fascinating listening to you talk so passionately about all these areas. I really wishing you the best in everything you're doing. Obviously, you're going to be extremely successful at it. Takes time, but nonetheless, it seems like you really have a great organization there and a great set of projects you're working on. For everybody that's going to be watching this episode on the YouTube channel or listening across the various podcast networks, you've been listening to Dr. Adam Freund, Principal Investigator, Calico Life Sciences, working to identify interventions that either slow or reverse aging. Adam, it was a was a pleasure meeting you. Thank you for taking the time to come on the show and talk for us, to us. And as we say, thank you for moving the human story forward and the equine story as well. It's been really a fascinating hour or so, and I really appreciate it once again.
All right, it's been a pleasure.